Welcome to A2 Selection and Evolution. At the heart of Cambridge A-level biology is one simple truth. Life is not static. Environments fluctuate, selection pressures shift, and populations must adapt or face extinction. In this unit, we move far beyond the basic idea of survival of the fittest. You will understand evolution as a highly quantifiable, dynamic process. Before we go any further, we need to lock down exactly what evolution means in the exam. It is defined strictly as the change in allele frequencies in a population over time. It is not just a change in physical appearance, it is a fundamental shift in the genetic makeup. We begin with natural selection and the types of natural selection. Environmental pressures dictate which alleles confer a survival advantage, and we will explore how this alters the distribution of phenotypes in a population. Stabilizing selection narrows the variation, favoring intermediate traits like human birth weight. Directional selection pushes the entire population toward one extreme, which is exactly what happens with antibiotic resistance in bacteria.
Next, we look at evolution and the evidence supporting it. To study evolution scientifically, we must measure it using the Hardy-Weinberg principle. By calculating the expected frequencies of homozygous dominant, heterozygous, and homozygous recessive individuals, you can prove mathematically whether a population is stable or actively evolving. And how do we prove these evolutionary pathways? Gone are the days of just looking at fossilized bones or anatomical similarities. Modern evolutionary biology relies strictly on molecular evidence. You will analyze differences in nuclear DNA base sequences, use mitochondrial DNA as a molecular clock, and compare specific amino acid sequences to map precise phylogenetic relationships.
When populations diverge significantly, we reach speciation. The absolute requirement for a new species to form is reproductive isolation. You must stop gene flow. We will look at allopatric speciation, where a physical geographical barrier splits a population. We will also tackle sympatric speciation, where new species arise within the exact same habitat due to ecological barriers, behavioral differences, or sudden genetic changes. The end result is always the same. The two groups can no longer interbreed to produce fertile offspring.
Finally, we examine artificial selection. In this topic, humans act as the selection pressure. We deliberately choose which organisms reproduce to rapidly alter the genetic makeup of domesticated species, creating disease-resistant wheat or high-yielding dairy cattle. But you will also evaluate the severe costs of this process. You will see how selective breeding leads to inbreeding depression, loss of hybrid vigor, and extreme susceptibility to new diseases because of severely depleted genetic variation.
By the end of this unit, you will confidently calculate allele frequencies, construct the exact logical sequences required for speciation exam questions, and evaluate complex molecular data. You are now ready to master the forces that shape all living things. Let's dive into the mechanisms that drive these genetic shifts.